Hello everybody and welcome back to the series on cancer biology. Today I'm going to talk about the first 5 hallmarks of cancer in detail. But before that, I would like to talk a little bit about the cell cycle and how it correlates to cancer. So we already know that a cell cycle has 2 phases. The first phase is called the interphase, followed by the next phase of cell division called mitosis. During interphase, the cell prepares for cell division, and during mitosis, the cell is divided into 2 identical daughter cells. Now this entire process occurs in a controlled environment just to ensure that the entire genetic material of the cell is passed on without any errors, and there are a number of checks that are involved that keep the integrity of all the contents that the cell has to replicate. The main objective to understand cell cycle is to understand why cancer cells are immortal. What you're seeing on screen is a typical diagram that represents a cell cycle. You can clearly see the interphase and mitosis represented by the yellow and orange parts. During interphase, the cell prepares for cell division by consuming nutrients and increasing the number of organelles and cytoplasm. Interphase is further divided into 3 distinct phases. The first and the last phase are known as gap 1 and gap 2, or growth 1 or growth 2 respectively. They are separated by the S phase known as the synthesis phase. During growth 1 phase, the cell basically prepares for DNA replication, and it ensures that everything that has to be copied or replicated is exactly the same way that it is supposed to be, and it involves a number of checks to ensure the integrity of the genome. During the S phase, the cell replicates DNA and doubles the genetic information that it contains. During growth 2 phase or G2 phase, the cell prepares for cell division, doing last minute checks to make sure that everything is in the right place. Now you might be wondering, what has all these phases got to do with cancer? Well, the answer is most of the cancers are due to errors in either the growth 1 or growth 2 phases or in the synthesis phase. And we're going to look at what these errors could be during our discussion as we move on. Now, after a cell undergoes cell division, it has 3 options to choose from. Either it can go back and divide again, or it can go and take a short break, or it can differentiate into a type and never divide again. This creates 3 categories. The first category is called labile cells, which means that these cells could multiply and divide again. The second, in which the cell takes a short break, is called stable cells. And the third, in which the cell can never ever undergo cell division, is called permanent cells. And we're going to now discuss them briefly. Labile cells are cells that after undergoing mitosis jump back towards proliferation and into the interphase. These cells continue dividing for a longer period of time, possibly for a lifetime. Some common examples of labile cells are your hematopoietic stem cells that make up your blood, or your epithelial cells that make up your skin. The cells that take rest for a short period of time after replication are called stable cells. This means that the cells have not exhausted their replicative limit, but at the same time, they will not continue replication unless they receive a stimulus or a special signal that activates them into proliferation. Examples of these cells are the cells of your liver and kidney. The third type of cell is the kind of cell that only undergoes cell division once. These type of cells cannot regenerate, they cannot proliferate, they have a fixed lifespan. An example of these cells are your neurons, or the cells of your skeletal and cardiac muscles. Now that we've covered these basic cell types, and we now know that each cell divides into 2 daughter cells, the question is, how many times does a cell divide? If you're going to search it on Google, you're not going to come up with a definite answer because there are a lot of factors that determine cell growth. The only important thing that you need to understand is that there is a limited number of times that a cell can divide. In the first video, I discussed about telomeres, which are at the end of the chromosomes, and every time a cell divides, these chromosomes shorten. Now, a cell can divide as long as it has a certain amount of telomere left. When a cell can no longer divide, it transforms into a senescent cell or goes into a state of senescence, which means that the cell will not actively participate in its duties. However, it continues to live and consume nutrients. This limited number of times that a cell can divide is known as the Hayflick's limit. Upon categorization, cancer cells can be called labeled cells because they're continuously replicating, which makes them immortal. And we're going to discuss how these cancer cells achieve replicative immortality in our first hallmark of cancer.